today. Mutations are and what types of mutations, etc. So let's start with that. So the first thing is why we need mutations or what are mutations? So mutations is defined as any heritable change in the sequence of your DNA. What is sequence of nucleotide? We call it as mutations. And the organism with mutation is called mutant, while the organism without mutant we call them generally as wild type. Now, why do we need mutations? Now, we all know that transfer of genetic material from generation to generation depends on how accurately or how precisely the information is passed on to the next generation. But what happens is the genetic material is not perpetuated with perfect fidelity. The genetic variation needed by evolution would be lacking and new species would not have arisen. That is why we need mutations. But at the same time, we should make sure that mutation is uh, in case it is happening in the germline cells, it would destroy the species, and in the somatic cells will destroy the individual. So there are basically two important sources of mutations, that is inaccuracy in DNA replication. Replication happens during that time, but error is there that can lead to some mutations, and the other one is chemical change to the genetic material. And these in response to certain chemicals, the DNA or the nucleotide might. So mutations can be genetic level or at the chromosomal level. Maybe you can say that it is happening in a particular gene, you call it as gene mutations. Then there is certain change in the chromosome, chromosomal level, we call it as chromosome mutations. Now, when you say when it is gene mutations, it can be a replacement of any nucleotide with another nucleotide, or it can be addition or deletion of nucleotide. Talk about mutations, change in the number of chromosomes. There can be a change in the number of chromosomes, change in the chromosome, both we call them as mutations. See the mutations were based on the change in genotype and phenotype. Mutations are of two types. They are point mutations, what are this? It occurs as a result of substitution of nucleotide by other in specific nucleotide sequence of the gene. Generally, these point mutations bring very little phenotypic change as compared to the other one, which is called as the perfect mutation. Point mutation can be transition or it can be transversion. Transition is the point mutation that occurs by substitution of one pyrimidine by another. Transversion is the point mutation that occurs by substitution of urine by pyrimidine or vice versa. You can see here it is a replacement of urine with urine transition. Transversion means if a purine is changed into pyrimidine, or if it is changed into transversion mutations. Based on the effect mutation can be silent mutation, sense mutation, and sense mutation. Silent mutation is also known as neutral mutation. Silent mutation, as I said, it is also known as neutral mutation. It is the mutation in which the mutated codon codes for the same amino acid as that of the original codon. So then we call it as the silent mutations. The missense mutation this mutation, what happens? The mutated codon codes for a different amino acid other than original. Now, since no amino acid coded by the mutated codon is altered or changed, the protein is also changed. Such kind of protein can be it can be less active or it can be completely inactive. What's Now the third one is nonsense mutations, mutation in which the altered codon is a stop codon or chain terminating codon. Now such mutation is called as nonsense mutations. So nonsense mutations causes incomplete synthesis, and incomplete synthesis is always non-functional. So here is the example. If it is coding, this is the example where there is the wild type is when when there is no mutation, you got it a normal protein. However, you have other few mutations that is missense, nonsense, and silent mutations. The other one is the friendship mutation, the second one. So it occurs as a result of addition or deletion of nucleotide in the sequence of the DNA. The addition or deletion of nucleotide causes shift in the reading frame of the mRNA. In an mRNA, each codon is represented by two bases, which is without punctuation. An insertion or deletion of a nucleotide changes the entire code. And this is the reason why frame-shift mutation brings greater phenotypic change than point mutation. So this is how there is an example here. So how this frame shift mutation works. So you can see here. Earlier we have CET like this, which is coding for histidine, because CET codes for histidine. So when it is read as a codon, it is reading as CET, which codes for histidine. Any insertion in between or any place that is going to shift the codon. So there is a frame shift here. You can see it is no more CET here. It becomes ACA, which codes for purine. And thus what we expected what this was histidine. However, now we have purine, and this incorrect amino acid may produce an incorrect protein. So now effect of the gene mutations, you can have neutral effect, you can have some harmful effect, or you can have beneficial effect depending on where the mutation has taken place. So if the mutation has taken place in a very important gene which has can be lethal to life, then we can call it as harmful. Now, if it gives a new version of the protein, we can call it as beneficial. And sometimes mutations might not have any effect. So that's a short review on mutations.